Okay, so here's our thick filaments in the center here, myosin. Thin filaments in blue at the top, right in between, that's actin. And then at the very end of myosin is this titin, this kind of curly protein. And titin is kind of just anchoring the thick filaments to the Z line. So titin is providing sort of the structural component for the sarcomere to be able to contract and lengthen. If titin were not there, myosin would not have sort of this anchoring point across which it can now drag actin, to the M line. So notice how titin anchors the thick filaments to the Z disc or the Z line here and provides a structural basis point at which it can then pull or drag actin in this direction. So if titin were not anchoring myosin, then there would not be enough structural integrity for it to be able to drag actin towards the center. All right. Another important protein, I think, is troponin. So troponin is a regulatory protein, but troponin is another kind of main star of the show. If troponin is not there, then calcium doesn't play its role in regulating the muscle contraction. So I think troponin is another important protein as well. And arguably, all of them are important, right? That's what I kind of wanted to highlight, is that each protein, back here, each protein plays a crucial role in putting the entire process together. And so they all play a specialized individual role that if they were absent individually, none of this would happen. None of this would turn on, none of it would work. So just to highlight the importance of all the players here that are, regulating this process. We left off looking at the process of cross-bridge cycling and then later on excitation, contraction, coupling. And I think we ended off describing here, which is where we said that the ending phase of the muscle contraction was putting that calcium back into the SR so that we could cease the muscle contraction. Specifically, if we take a look at the structure of actin, when the calcium is absent, troponin drags tropomyosin to cover up the binding sites on actin. Okay, the binding sites for myosin. And then when calcium is present, the opposite occurs. Troponin, which is where the calcium binds, drags the tropomyosin, that thread-like purple protein, off of those binding sites, exposing those myosin binding sites, and then myosin can engage and then contraction can happen. Okay. So that is where we left off. We're going to continue on here and talk a little bit more about the details of how the T-tubules allow the action potential to spread and how the spreading of the action potential results in calcium release from the SR. So the gating mechanism of the sarcoplasmic reticulum is such that there is a coupling of the T-tubule. So the T-tubule is this green space, this invagination or this tunnel that runs through the depths of the muscle. And the T-tubule is coupled by a ryanodine and a DHP receptor to the SR. So here's the surface of the T-tubule. Here's the SR in blue, right? This is where the calcium is going to be stored. And what is coupling these two structures together are two receptors or two gating structures. So one is the ryanodine receptor, which is the smaller green structure closer to the SR. And the other is the DHP receptor, the dihydropyridine receptor. And that's the purple structure that's tethered to the T-tubule side. So these two structures help to pass that electrical energy and help to act as a gating mechanism of the SR such that when the voltage is right, the DHP receptor signals to the ryanodine receptor, and the ryanodine receptor is acting not only as a voltage sensor, but also as a channel. we've seen other receptors that have acted in this way. So when the electrical current gets passed from the T-tubule to the purple structure, which is the DHP, and then from the DHP to the ryanodine, which is the green structure here, then because ryanodine receptor is also a channel, it's going to open up. And in doing so, in opening up that channel, calcium can now begin to leave the SR in the blue space here and move outward into the white space, which is communicating into the general cytoplasm of the muscle fiber. Okay, so in doing that, calcium will now leave the SR, flood the cytoplasm, engage with troponin, and regulate that muscle contraction by way of uncovering those troponin, those actin binding sites for myosin, right? The binding sites on actin for myosin. The other piece is that once calcium leaves the SR and begins circulating in the cytoplasm, it's going to induce more of these channels to open up. All right, so the more calcium we have, the more we can induce opening up of further channels. And then there becomes a point where the cytoplasm is so full or so, there's so much calcium in the cytoplasm that calcium can also be a trigger to induce closing of the receptors and the channels as well, right? The ryanodine receptors. Okay. Now at the bottom here, we can see what's happening with the schematic. So here's the action potential traveling down the T-tubule. Remember, the T-tubule is on this side of the membrane. It's going to signal the DHP receptor, and then that is coupled or tethered to the ryanodine receptor. So when the voltage changes, the DHP tells the ryanodine receptor to open up that channel. Calcium can leave the SR, which is presumably on this side of the receptor, and then go out into the cytoplasm on this side of the receptor, and then flood the cytoplasm, and then have its role in muscle contraction. All right. Now, again, just to reiterate here, in order to terminate muscle contraction, calcium must leave troponin, and then it must also be pumped back into the SR. It must leave the cytoplasm and go back into the SR where it is stored. So it's really important for calcium ATPase pumps to be present on the SR membrane as well. And that helps to transport that calcium from the cytoplasm back into the SR so that we can cease that muscle contraction. Otherwise, we can get into states like tetanus, right? Which is uncontrolled muscle contraction. And so that, not tetanus, excuse me, tetany. Tetanus is a very different thing. Tetany, which is an uncontrolled muscle contraction, and then that can be unhealthy for the muscle as well. So it's really important to cease that contraction by way of the SR pumps, calcium ATPase pumps, putting that back into the SR.